genes. As we have just studied that genes are the genetic material which codes for the characteristics or traits to be passed on from parents to the offsprings. Now genes which code for pair of opposite traits or characters are known as alleles. To represent this, we will be using the upper hand of alphabet T to give tall and the lower hand to tell us about DAF. If we will be using T and D, it will be difficult for us to further understand the characters and to understand that these opposite characters or traits are present on the same gene or are coded by the same gene. This is the representation which tells us about the genotype. When we write like this tells us about the genotype, and when we say tall and dark, it tells us about the phenotype, that is the physical appearance. I see a tall plant, I will say this plant is tall, that is I am describing the phenotype of that plant. Now, what Mendel did, Mendel said that this F1, after hybridization, the gametes formed were this, which is heterozygous in nature, but these gave, this is homozygous in nature. Now, in F1 what we observed that this, though the genotype was this of the plants, but when we saw the phenotype, they all were tall. Thus, this T, the capital T, means the tall character was dominant over the dwarf character, that is T is dominant over this dwarf character, and this is known as recessive. Now, this was the case of F1. Once then we reached to F2 stage. In F2 stage, we found that recessive trait is expressed but without any blending. That is, there we saw that it was the ratio of 3:1. One dwarf plant was observed, but there was no blending. That is, there were 3 tall and 1 dwarf plant. There was no plant of medium height. Now, how this dominant or recessive character or trait is passed? When parent plants undergo meiosis to lead to the formation of gametes, alleles, they separate or segregate. That is, one allele, that is one particular trait, it can be dominant, it can be recessive, goes to one gamete. This segregation or separation is random, hence there are only 50% chances that either tall trait will go to the gamete or the dwarf trait will go to the gamete. To understand this further, a scientist named as Renald C. Punnett gave us this Punnett squares, in which we write down the gametes on the either ends, and this is used to calculate the probability of genotype of the F1 and F2 progeny. You can see here in F1 a tall plant is crossed with a dwarf plant. So, 2 tall characters are here, 2 dwarf characters are here. What we found that all tall plants are observed, but their genotype was tall dwarf. Hence, it showed that tall was dominant and dwarf was recessive. When we did selfing here in F2 generation, these were the gametes. We found that 3 tall and 1 dwarf plant is formed. Here you can see the dwarf plant is homozygous in nature, and it was further seen that when the DAF plant from F2 is further cultivated, it was found that it produced DAF plants in F3 as well as F4 generations. So, alleles are the one which are having opposite traits. These traits are passed on to gametes during the process of meiosis. Alleles separate, segregate, and they are passed on to the gamete. That is one allele to one gamete. This is a random process, so chances are 50%. Mendel said that genotype is this representation and phenotype is the physical description when we say a plant is tall or dwarf. Yes, on the other hand, we may say that genotype and phenotype are not similar to each other. In F2 generation, as we observed in Punnett's scare, 3:1 is the phenotype. That is, 3 tall and 1 dwarf plant was observed. But when you see the genotype, it is one is to 2:1. These were the genotypes, but the phenotype was 3:1. Hence, one can say that yes, alleles are transferred but randomly without any specific rate of transfer. Further, we can use Punnett square to calculate the probability of the genotype of the given plant, and we can use this Punnett square to understand the phenotype and genotype. That is, phenotype and genotype are not related to each other. To understand the genotype of tall plant so observed at F2 generation, Mendel proposed a test cross. In this test cross, the tall plant from F2 generation is crossed with a dwarf plant, and the progeny so obtained is used or is studied to understand the genotype. Now, after studying these crosses, Mendel gave us 2 laws of inheritance, law of dominance and law of segregation. In law of dominance, he stated 3 things. First, characters are controlled by discrete units which are known as factors. These factors always are present in the form of pairs, and if these factors are dissimilar in nature, one will be dominant and the other one will be recessive. So, according to law of dominance, characters or traits are controlled by factors. Factors are present in pair, and if factors are dissimilar, one is dominant, other one is recessive. Law of segregation. As we know, no blending of alleles is observed at both F1 or F2 generation, but one of the character is absent in F1, but when you see F2, both the characters or traits are recovered at F2, whereas in case of F1, one character was absent. So, when the homozygous gametes are there, the alleles are equally distributed. That is, only one trait or character is present. But in case of heterozygous gametes, both the traits are present in equal proportion. As we know, it's a random division, so there are 50% chances, that is 50 of them will have tall and 50 of them will have dwarf. So this is law of segregation, which states that no blending of alleles is found at F1 or F2 generation. Both the characters are recovered at F2 generation, whereas one character was not present at F1 generation. And further, when gamete formation is there, homozygous parents gives gametes which have only one character or one trait, whereas heterozygous gametes have both the traits or characters present in equal proportion.